There are three different classes of membrane receptors, including ion channel receptors, G-protein coupled receptors, and enzyme-linked receptors. This video will explain the signal transduction cascade of only the G-protein coupled receptors. Also, the G-protein coupled receptor in this video will activate the second messenger cyclic AMP in order to accelerate metabolic activity of the cell. But keep in mind that G-proteins can activate other second messengers, like cyclic GMP or calcium ions, to elicit different cellular responses. Since G-proteins act through these second messengers and indirectly affect the membrane potential, they are known as metabotropic receptors. The signal transduction cascade in the G-protein coupled receptor begins with a ligand binding to the receptor protein. A ligand is a substance that binds to the receptor to communicate a signal. It is the first messenger. The ligand in the G-protein receptor can be a neurotransmitter or a hormone. Some examples of neurotransmitters and hormones that bind to this type of G-protein receptor include norepinephrine, epinephrine, calcitonin, and parathyroid hormone. The binding of the ligand to the receptor causes activation of the G-protein complex, which is attached to the receptor. The G-protein is heterotrimeric, meaning it is composed of three subunits, the alpha, beta, and gamma subunits. G-proteins also bind to GDP. When activated, the G-protein converts guanosine diphosphate, GDP, into guanosine triphosphate, GTP. With GTP attached, the alpha subunit then detaches from the other subunits and binds to an enzyme. In this case, it is adenylyl cyclase. Inside the inner surface of the plasma membrane, adenylyl cyclase then converts adenosine triphosphate, ATP, into cyclic adenosine monophosphate, thus increasing the levels of cyclic AMP inside of the cell. Cyclic AMP is the second messenger that typically activates a kinase. Here it is activating protein kinase A, pKa. Kinases phosphorylate proteins in the cell, which means they attach a high-energy phosphate group to proteins and change their structural conformation. These phosphorylation reactions by protein kinase A can mediate both short and long-term cellular responses depending on the proteins that they phosphorylate. Ultimately, the cellular response in this example is an acceleration or increase in the metabolic activity in the cell. In order to stop this cellular response, an enzyme called phosphodiesterase, PDE, can inactivate cyclic AMP by converting it back into AMP. That concludes the signal transduction cascade for G-protein coupled membrane receptors using the second messenger cyclic AMP to accelerate cellular metabolic activity. Thanks for watching, Dr. Pirtle.
Yeah, get it, girl. Oh, yeah. Oh. So much sexy happening. Yeah. Oh gosh, it's too hot. It's too hot.